Okay, hello everyone. So chapter 3, gene mutations, and we're going to have a look at sickle cell anemia. So first question, what is sickle cell anemia? And more specifically, what is anemia? So anemia is when a body does not have enough oxygen. That means the organs are not getting enough oxygen. Sickle cell anemia is one type of anemia, and we're going to try and explain why this causes anemia. Okay, so number 1, sickle cell anemia. Observe and make a drawing of the two following slides: human normal blood smear, human sickle cell anemia blood smear. So I've got a picture here where we have someone who has sickle cell anemia. So note that in this case this person has both types of red blood cells. If I enlarge this, we can see, if I circle out using black, so we've got here, this is a sickle cell and this is a classic cell. So this one is nicely roundly shaped, that's a classic shape for a red blood cell. This one here is slightly sickle shaped, this one here as well, and this one here as well, as long as it's not a nice circular shape, so this oval and these sickle shapes. Here are two types of red blood cells. I'm going to switch back to the lesson. So I'm going to get this off. So if we are to draw them, we're going to have, for a classic red blood cell, a nice circular shape, and for a sickle cell, a sickle shape. Just in terms of size, a classic red blood cell is roughly 7 micrometers, between 7 and 8 micrometers. Okay, so this is what we can observe with the microscope: two types of red blood cells, a nicely circular shaped one and a sickle cell shaped one. Okay, let's move on. So the question is, can this have an impact on the organism? Well, the answer is yes. When you have classic round blood cells, round red blood cells, they circulate quite normally through these tiny vessels which are called capillary vessels. However, if we have sickle cell shaped ones, they're going to tend to pile up and block the passage. So they pile up because of this different shape, they pile up and they block everything. I have a video here that might be a little bit more telling. So I'll get that off. So you can see here that it blocks the vessel. These sickle shaped ones, they block the passage for the other ones. Okay. So why do we have sickle cell shaped red blood cells in the first place? Well, this is due to genetics. The answer is that this particular type of sickle cell anemia is a genetic disease. If we're talking about genetics, we're going to obviously have a look at DNA. So here we have the DNA sequence for beta globin. So we're going to do a recap. What is beta globin? So I'm going to go and get another slide. So in red blood cells, there's something called hemoglobin. this protein here is hemoglobin. Hemoglobin is made of 4 polypeptides. 1, 2, 3, 4. We've got 2 alpha globins and 2 beta globins. In the center of a globin you have a heme which has an iron atom, or iron to be more precise, Fe2+, and the fact that there is this iron will enable oxygen to be carried. So these hemoglobins with their hemes, they are going to be able to carry oxygen. This is another representation. So with the, you can see the alpha lexes. So again, we've got 2 betas and 2 alphas. Last thing, in one red blood cell you're going to have thousands and millions of these hemoglobins which carry oxygen. So these are vital to our body because they carry oxygen. So beta globin is one of the 4 polypeptides found in hemoglobin. Okay, so let's have a look at this DNA sequence. So we have TGA, GGA, CTC, CTC. This is for a normal beta globin. And if we look at a mutant beta globin, we've got TGA, GGA, CAC, CTC. And we can see that here there's a difference. The difference is that we have a thymine here and an adenine there. This is called a mutation. Thymine has been replaced by adenine in the mutant beta globin. Will this have an impact? That is the question. It will. We saw in the previous chapter that DNA is going to be transcribed into mRNA by DNA polymerase, and then that the mRNA is going to be translated into an amino acid by the ribosomes. So let's have a look at what this is going to lead to. So what we have here is the template sequence. The template sequence is going to be used to build the mRNA. So if I have CTC on the DNA, therefore on the mRNA I'm going to have something which is complementary. This is the Chargaff laws. So if I have a C on the DNA, I'm going to have a G. If I have a T on the DNA, I'm going to have an A, and the other C is going to be a G. So the 3 nucleotides are going to be GAG, GAG. Whereas for the beta globin, we're going to have G. So remember, in mRNA, instead of using T's, fine when we use uracil, U. So G, U, G. So we've got a difference. We've got GAG versus GUG. So next thing we're going to do, we're going to have a look at the genetic code. Okay. I'm going to get rid of. So I'm going to take these off. Okay. So GAG. So I look at the first letter G. So that means I'm going to be on this big line. Then A. So this is going to be GA. So I'm in this box now. G A G. This is glue. If I have a look at this document, glue, GLU, is, where is it? There we are, glue. Glutamic acid, this is one type of amino acid. So if I switch back, we're going to have, actually, we're going to start completing what we have at the end. So for normal sickle cell anemia, the DNA is CTC, which means the RNA is GAG, G-A-G. We saw that for the person who has sickle cell anemia, it's C-U-C, and therefore it's going to be CAC, and therefore it's going to be GUG. Okay, so the corresponding amino acid is going to be glue. So glue, we said that glue is glutamic acid. Equals glutamic acid. Okay. So what about GUG? So I go back here, G, so it's going to be this line, U, so we're into this box, and G, so it's going to be VAL, and VAL is VAL, where is it? Aha, valine. So there we go. We're going to add VAL equals valine. That's good. Valine. Okay. So first comparison: CTC, GAG, glue, glutamic acid, CAC, GUG, valine. There we go. Okay. So we've got our sequences. So this is going to be the amino acid glue, GLU. Now I can write. It's not very big, but I can write. So GLU and val. It doesn't seem to be working as well as I expected. Never mind. So now what we're going to have a look at. So amino acid of the normal beta globin, it's going to be val. I'm just going to isolate it. Oh, sorry, that val is not going to be for the normal, it's going to be for the mutant ones. So I'm going to try and glue this there. There we go. So, val. How do I put it in front? There we go, val. And we said that's glue for the normal one, so glue is here. I'm going to take glue and I'm going to place it here. Again, put that there. So we can see that there's a slight difference. So we've got the COOH group, we've got it here, we've got it there, we've got the H2N group. However, we've got another COOH group on this one, and we've got two methyl groups. So this is, you need to imagine that this is a carbon with 3 hydrogens, CH3, and this one is also a carbon with 3 hydrogens, CH3. Okay, moving on. This is going to have an impact because you know that from S6 we consider the primary structure, which is just the polypeptide. Then the secondary structure is how these polypeptides have a certain shape, local folding, and then the tertiary protein is how the polypeptide, how one polypeptides with the alpha and the betas bind together. When you have, for example, CH3 groups, this is going to create hydrophobic interactions, meaning that this part here is going to be closer to this part there because of these groups. If these groups weren't here, the shape, the three-dimensional shape of the polypeptide will be different. And we've just seen that here we've got CH3. So this mutation provokes a change in amino acid, which is valine, and valine has these CH3 groups, creating hydrophobic interactions. So what we can do is add these to your final document. Glutamic acid and valine. The reason we're doing this example in detail is that you need to know this example for the back. You only need to know one detailed example of mutations, and it's this one. And the good thing is that you will all have the recap on this single page. So impact on the tertiary structure, well, no hydrophobic interaction. In this case, there is going to be a hydrophobic interaction. Okay, I'm going to cut the video here just to do a first test, and I will continue in the next video. So this is going to be a first test, see if it works. Goodbye.